So we will figure that out. But I guess the bottom line is, don't worry about your homework since you couldn't, that part of it anyway, you couldn't do it.
Has anyone been able to get onto the Tree of Life website?
Yeah, I've been able to get on it.
So anybody?
Yeah, I got on
Yeah, it was working last week. Is that when you did it?
it. Yeah.
I did it last week.
So everybody who was able to get on successfully, that was last week.
Mm-hmm.
Nothing after, what, Sunday?
Yeah, I tried on Sunday and it was down.
It was down. Okay, all right. All right, well, there you go. Now we've nailed it down. Thanks. Yeah, always feel free. It's important to let us know these things. So thanks for doing that. You shouldn't be shy about that. So we'll have to readjust. Maybe we'll delay that part of your homework a week. Or I mean, if you already get it, you can turn it in. But doesn't sound like too many people did before it crashed. So maybe it'll be back up this week, or we'll have to find a way to replace that. Okay. Great, thank you. Well, look what time it is. It's that time. Time to begin. So, an announcement for lab. Bring your computers to lab this week because you're going to be doing some analyses that will require your computer, and they're fun. And if you have it, you'll be able to get the program and do the analysis. And we're also going to decide this week if we're going to order these books, the Kastner et al. Book. So take a look. We're going to make it. You'll make a decision in lab this week. Come.
We're supposed to meet at Rose to go to the herbarium, not at the East Hall.
Right. So except for one lab. I think you guys know that, right? You're meeting in Rolls, except
5 o'clock.
5 o'clock
5 o'clock Thursday.
Thursday, and we can make that announcement again Thursday. But yeah, except if you're the Thursday evening lab, go to Rolls. Okay. So here's where my lecture is today. It's from chapters 1, 2, and 5. And I'm going to, eventually stop posting the complete PDFs. I mean, if you're coming here, that's great, then you can take notes. But what I don't want is people to say, I don't need to go to lecture. I'll just look at the PDF. So I wanted to keep posting the PDF, but I probably will not everything will be on there. Yes, giving you an incentive to be here. All right, one thing we're going to start today is just trying to get you to make sense out of the green world that's out there and start paying attention is what's called the plant of the day. And this is one that's pretty common around town, around Florida, around the campus. And these are wet just because of rain, don't worry. This is, the common name, as you can see here, is Cape Plumbago. It's widely planted, as I said. It's a plumbago is the name of the genus, Plumbago auricula. Auriculata is the name of the species. It's in a family, Plumbaginaceae, that we are not going to cover. It's in a group that we're going to cover. They're the Caryophyllales, an order that is just full of cool plants. So with something like this, these plants of the day, which I'll pass around, what we're trying to get you to do is notice plants. There's a bunch of dead stuff on this one. So just pass this around, and on the first test, it will be, how many plants of the day can you list? That will be extra credit. All right? I got a little bit more in here. This is not such a good looking bag, that one. As you can see, it's sort of a big herbaceous, almost not quite shrubby thing. That's a real tree for scale. So they often plant this as almost like a hedge in places, though it's not quite that robust. These beautiful flowers. So people just call it plumbago, okay? And I would accept that. If you want to call it Cape Plumbago because it's native to the Cape region of South Africa, that's fine. But sometimes I'll bring in one, sometimes two lectures. It's up to you. If you want to pay attention and learn them, that's fine. If you don't, that's fine. But my idea is you're going to see, if you walk around campus today, you're in all likelihood going to run into this plant. It's in full flower. You say, whoa, is that the Cape Plumbago I heard about in lecture today? Eureka, and you'll be happy. Okay, I'll keep doing this for a while. Possible test questions. Why is the classification of Linnaeus considered artificial? What is meant by that statement? Yes?
It didn't take into account evolutionary relationships.
Perfect. That's right. It didn't take into account evolutionary relationships. It was simply for convenience, right? People were discovering all these organisms out there. How do we group them? How do we catalog them and put them and make sense of all this diversity? And Linnaeus was one of the first great classifiers, but he did not think in terms of relationships. He just said, oh, yeah, you got 5 stamens. Oh, you got 5 stamens. I'm happy. You're together. Just like we did last time jokingly. You have a hat. You have a hat. You must be closely related. Okay, stuff like that. All right, so what happened in 1859 that influenced systematics? Origin of species. Today we want our classifications to reflect, this is an actual test question, 2 blanks, so you know there are 2 words. We want our classifications to reflect evolutionary relationships, okay? And what is a clade or monophyletic group?
All the descendants of a common ancestor.
There you go. An ancestor and all of its descendants. Not an ancestor and some of its descendants, ancestor, all of its descendants. Are we getting this down? So already, how many points do you think I've given you in the first test? A lot. And this is the kind of stuff that Pam and I will be asking you, and they will reinforce in the laboratory. Okay, this is an actual tree from your textbook. Here we have, these both are what are called synapomorphies for the clade of 2 and 3. 9 is a synapomorphy for 1, 2, and 3. These characters 1, 2, 4, 6, 7, and 8 would all be unique to 3. That's called an autapomorphy. Aut single 1. Okay, so there you go. This is just a review. Now, I said that sometimes these things are relative. We even talked about this, that if suddenly we found 2 more species, 4 and 5, and they were, close relatives of 3, then all of a sudden these characters are now, if they're all in those 3 species, they're synapomorphies. They were autapomorphies, but now those 3 species all have them, and now they're synapomorphies. Got that? They're kind of, these things are relative based on the discoveries that take place. All right, we talked about the magic scissors. You do not want to cut up your textbook now, just trying to find out if something is a clade, but this is the way to do it. If you can snip out a group with 1 cut of your scissors, it is a clade, right? If it takes 2, it's not. So a monophyletic group, we've already said a descendants of a common ancestor. We're going to introduce a new term, which would be represented by the reptiles. Because if as traditionally you recognize, they did not include the birds, but the birds should be in there. So if it takes 2 cuts, as it would, to get out what were traditionally considered reptiles, that's not a clade. So this is something we call a paraphyletic group. An ancestor and some of its descendants, okay? So in this case, there's an ancestor of all of these, but if we only consider, or try to consider that as a group, that doesn't work, right? That's a paraphyletic group. And here is what your reaction should be to paraphyletic groups, okay? From this day forward, you should be, this is The Scream, a famous painting, and that's how you should think about paraphyly. It is bad. We do not want to name groups that are paraphyletic. Now, we'll come back to this. Some people still try to do this. And you say, wait, why would you do that? Okay, all right, an ancestor and some of its descendants. Okay, now another term that's important, and you will see this, this is all part of our phylogeny 101, background information. Notice that this character 2, whatever it is, we're not telling, we're just using numbers, that little slash mark, is on the branch to species 3, but it's also over here on the branch to species 1. Everybody see that? All right, this is something called homoplasy. Homoplasy, where you see the same state is in 2 places. This is a new term. Remember, I'm trying to do this gradually. We're going back, reviewing, but now I'm throwing in some new terms. So homoplasy, this is a similar feature due to parallel evolution or reversal. You know these terms. You know these terms. You just don't know you know these terms. You probably have heard them somewhere. Parallelism is the separate origin of the same character state in two or more organisms. Okay, parallelism. Now, you can also get homoplacy by reversal. A character originates, it evolves, a derived character state. But then, somewhere down the road, it reverses back to the ancestral state. Imagine a lineage of plants. And the ancestral state, white flowers. Species, species, species. Some turn in, they get red flowers. Species, species, species. But then one reverts to white flowers. That is a reversal, okay? So what causes homoplacy? Parallelism and reversal. Parallelism and reversal. Homoplacy just happens, folks. This is not something bad. It's just something that we have to be aware of as we build and relook at phylogenetic trees. Homoplacy happens. Do these have wings because they got them from a common ancestor?
No.
I do not think so. All right. Now, the animals. This is a great example you'll see in class of plants. This is a cactus. This is not a cactus, but it sure looks like a cactus. But two very dry adapted lineages of flowering plants, not at all closely related, as you will see. We'll get into this, so how you can tell them apart. But that, similar adaptations, a whole series of parallelisms, okay? Now, this would be what I would say, and this is my license plate. Everybody get that one? Read it. Homoplacy happens. Get over it. It's a cool aspect of evolution. I don't really have that as a license plate, but I would like to have it as a license plate. I think it would be cool, all right? So you would understand. Everybody would see this. Yes, of course, it's just part of evolution. Now, okay, everybody good with those terms? What are the new terms we introduced? We introduced homoplacy. Everybody got that? Homoplacy, I know what it is. Paraphyly, a paraphyletic group. Do we like paraphyly?
No.
No. Okay, so, as you remember last time, we were building trees. And we said we knew what the ancestral state would be, and that some character states might be derived, and those changes we use to build trees, as in this example from the book. Now here is the ancestral suite of features, which we call simplesiomorphies, and then here's how they changed across this tree. So the question is, which we didn't discuss, but now we are going to discuss, and now you'll know the answer, is how do we infer the ancestral and derived states? How do we know that? I mean, in any group, this may have happened thousands, millions of years ago, okay? How do we know, unless you were here and you weren't, of course, millions of years ago, to know what the ancestors were? So what we do as we build trees, again, this is part of our course or our crash course in building phylogenies, building trees, is how do we determine what the ancestral states are, how do we infer them, and what the changes are, what's derived. So we do that by something that's called rooting. Rooting. We're going to determine which changes occurred relatively more recent and which occurred deep in the past. Think of a tree, just where it's a tree, branching tree. We want to know what's down at the base, at the root of that tree. We're going to root our tree. We're going to root the features of that tree. Everybody get the term? Use it in a complete sentence. I want to root my tree. I'm going to root the changes so I know what occurred, when they occurred. All right, so this gives our state changes direction. It tells us which are ancestral, which are derived, because that's key in building trees, right? We have to know what the ancestral state is and the derived state. And so how do we do that? This process of determining what is ancestral, what is derived, we're polarizing the changes, just like you would go, the polarity on a battery. You're going from minus to plus. It's the same thing. We're just trying to figure out the directionality of the change. New terms, but they make sense, right? Root. We're going to root our tree. What's ancestral? What's derived? By doing that, we're polarizing the changes. So how do we do that? Since we weren't here when a group evolved, say green plants, which are, what, 1 billion years old? Anybody here then? No. Okay. We use an outgroup. We'll use an outgroup. What's an outgroup? So we root by using a relative of the group that we're going to study. Okay, what is a close relative of our group? And we'll look at it and its features, and that becomes the way that we root our tree, by using an outgroup. We use a relative of what we're going to study. Okay, everybody understand that? You have to find what you think is something related to your group, which is not always easy. There's some background that goes into this. And then you're going to use that to root your tree. That's going to be your outgroup. Let's give you an example from your textbook. Okay. Here we have 3 imaginary species, 1, 2, and 3. Beautiful flowers, right? So that's the group you're studying. That's the group you're going to study. Now, how do I build a phylogeny for these 3 trees? How do I'm going to assemble characters, I'm going to make states for those characters, but the question is, how do I determine the direction of change? How do I polarize those changes? How do I find out what's ancestral and what's derived? And we're going to do that using an outgroup, something that, okay, I know a bit about these plants. This is something that's closely related to the group that I want to study. You know, in the real world, you'd probably try multiple outgroups, because a lot of times, do I know, do I not know, what's the best outgroup? We're just using one, okay? Everybody got that? A close relative of our study group is the outgroup, and we're going to use that to polarize changes, to look at the direction of changes of our states. So this is from your book. We're going to look at these characters: roots, stems, leaves, venation, all the way down through flowers, just like we did last time, right? We have characters, and then we have states of those. Here are the states. For the example, the flowers, they can be in groups of 3 or just 1, okay? The leaves, they can be alternate, that means, or they can be right across from each other, opposite. We built a matrix of these characters and states. Now, what we're going to do is use our outgroup, compare the in-group to the outgroup, determine the directionality of these changes, and build a tree. Are you ready? Right from your textbook. Okay, let's start with the flowers. Now, the outgroup, notice, has its flowers in a cluster of 3. Pretty obvious. All of the in-group species have a solitary flower, okay? So our character is flower. The states are clustered in a group of 3, 1, right? So using our outgroup, that is a synapomorphy for our in-group, the species 1, 2, and 3. We are building a tree. / character change right there. You see how that worked? That's not hard. That's not hard. And how did we do that? How did we determine the direction of change? We used an outgroup. What was the character of the outgroup? We're going to call that ancestral. What the in-group has is derived. Good? Good? Shall we keep going? Let's pick up. This is fun. This is what phylogeny is all about. Let's look at the leaves. Okay, we have 2, that's the character, 2 states. Alternate, opposite. Not tough, not tough. Now, here are our beautiful imaginary plants. Now, what's the state for the outgroup? The leaves are alternate. Look at our in-group. One of them has alternate leaves, like the outgroup, but the other 2 have opposite leaves. Now, if we want to build a tree, which we do, that's what we're doing, that change right there to alternate leaves unites those 2 species. The outgroup and one of our in-group have opposite leaves. There was a change here to alternate leaves, which unites 2 and 3. Everybody see that? So that means this is using our terminology, what's a shared derived character? This is a synapomorphy for 2 and 3. Our tree is building. We can go through everything compared to our outgroup, the 3 species. There's our little matrix. And eventually we end up with this as our tree, taking all of those characters and state changes. These are both synapomorphies. These are all autapomorphy. We have one example of a homoplasy because it occurs in two places. We built a tree. How did we do it? We used an outgroup. You have to have an outgroup. It really helps to have an outgroup to infer the direction of change. Good? Okay. Now, the history of tree building. Okay. How do you decide what tree to pick? Because you're going to see that if you have a bunch of characters and states, there may be several ways to build a tree from those characters and states. So the way that we use, or we used first anyway, is something called parsimony. And it has a great history, an amazingly cool history. And it's very easy. Parsimony, the law or principle of parsimony, is that the simplest hypothesis is the best. The simplest solution that explains the data is the best. This is the principle of parsimony. Now, this actually was made famous by William of Ockham. How many people have heard of Occam's razor? He was really a theologian, English, 14th century, and he was called Occam's razor because he used the principle of parsimony when he debated other people who were logicians to cut them to shreds. His argument was, that's way too complex. The simplest argument is blah. That's what I prefer. Okay, so the simplest explanation to explain the data is the best. That is the law or principle of parsimony. We are going to use that to pick trees. I guess the way to think of Occam of razor, he was sort of the, I know Mr. T is kind of getting old, but he's sort of that kind of guy of his time, I pity the fool who uses a complex explanation to explain the data. Notice the similarity of weird haircuts, so they have a lot in common. Similar types of attitude. Look at those smiling faces. Anyway, let's use it in practice. Here we have a data set with characters and character states. This is, you're looking at frogs, birds, crocodiles, kangaroos, bats, humans. And here are your states, a number of characters and states here. You don't really have to look at all of them unless you really want to. That's not the point. But you could put those, take those data and make a number of different trees. Here's one, here's another one. All right, how do I determine which one to pick using the principle of parsimony? The simplest is the best. So what you do is you go through and each one of these is a change. You count them up. How many changes on this tree? How many changes on that tree? The number of changes to build a tree is called the length of the tree, okay? How long is a tree means how many of these little steps or changes that I need to make it. All right, so using those two, tree 1 and tree 2, for no points right now, but maybe on the next test, which is the tree you would pick based on the principle of parsimony?
1.
What? Which one?
Top one.
1, the top one. Yes, you say you make the call. Yes, tree 1, because it is only 7 steps in length, okay? Takes 7 changes. This one takes 10. There's a number of different examples of homoplasy. It's too many steps, too many added steps to make this tree versus that tree. We go with the top tree using the principle of parsimony. Yes?
Sorry, how do you count the steps? Is it usually
Each one of these is a step.
Okay.
So when you build your tree, just as we did in that first example, you're going through, there's a number of ways that you can arrange your characters and character states, and you say, how do I pick? How do I, at the end, you can generate lots and lots and lots of trees. We'll go over this later. So how do I pick? How does the computer pick now? Because this is all done, and you will do this with computer programs. You actually count up these changes, and with big trees it can be thousands, millions of changes, and you pick the one that is the shortest, okay? Each one of those changes of a state is a step. Got it? Good on that? Okay, so the simplest one is the best. So when did this method of building trees come about? Really what we just gone over, this whole idea of using characters and character states to build trees in a very logical fashion, and then using something like parsimony to pick the one that we think might be the best tree as we generate hypotheses, was not done until really the 50s. And this is the guy, did it, Willy Hennig, an Austrian entomologist. He studied incredible history. The guy, what a life. Entomologist, ended up on the German side of World War II, but all he really wanted to do was study insects. After World War II, he took his theory, and this is the stuff he came up. Does that look familiar? Right? That's from Hennig. This look familiar? He's building trees using characters and character states, and that traces to Hennig. So if Darwin were around, remember we looked at the tree from the Origin of Species in 1859, what took so long? A century, really, to come up with real ways of making trees. So until Hennig, and even after Hennig came about, this is how people built trees. This is Haeckel's, one of Haeckel's famous trees of life. You're just drawing out how you think things are related based on your maybe a lifetime of experience. Here's a botanist, Bessey. He had this famous organization of how he thought some of the flowering plants are related, and it kind of looked like a cactus, so it became known as Bessey's cactus. But this is not a rigorous way of building trees. This is just somebody who knew a lot about the organisms. Here's how I think they're related. Here's how I think they evolved. But what we want to do is this kind of stuff, modern tree building, okay? How are we going to do that? We're going to use an outgroup. We're going to use characters. We're going to use character states. We're going to use actual ways of building trees and putting them together. Okay, so phylogeny terms. You'll see a cladogram, but it means the same thing as a phylogenetic tree. I don't care, you can use either one you want. Okay, it means the same thing. We have character and character states. We've given you an example. There ones we started on that last time. There's a number in your textbooks. This is one from your textbook where you're using appearance, what we call morphology, to generate a bunch of characters and character states. Everybody good with that? All right. There are tons of characters and states out there. Leaf characters. Think of all the features of leaves that could be used. Hairs on plants, all these different hair structures. The problem with these, and I'll say more about them, is that they can be very, very hard to compare across different organisms. People have even used pollen grains, chemistry, all kinds of features. So it was there were lots of ways to do this, but it was very challenging until something came along in the last few decades that really caused a revolution, and that is the use of DNA. Now, sometimes you'll hear people talk about molecular systematics, using molecules to study relationships. Sometimes early on people used chemical compounds, RNA, enzymes, but today when people say molecular systematics, they mean DNA. DNA is the tool of choice for building trees. I have given you simple examples using morphology, appearance, because those are easy to go through and understand. But if you were really going to build your own tree, if I were going to say, by the end of next week, your homework assignment is to build a phylogeny of X, of a group, here's the plants, and you really wanted to do your best job possible, you would use DNA. Okay? And I'll just spend a little bit of time on this because it is amazing what you can do with DNA. So let's just use our same terms. It's very easy to understand the states in DNA. So here we have our 4 species again. Maybe 4 is the out group, 3 are in group, it doesn't really matter. Each of these is a sequence of DNA, let's say from the very same gene, very same segment of DNA. That means each one of these is a character. That position on that gene is a character. That is a character. That's a character. Whoa, we're getting a lot of characters, okay? Especially when you think that, well, there may be 20 to 30 to 40,000 genes in a plant. Whoa, that's a lot of characters. But what are the states? Very easy with DNA. They are always, unchangingly, G, A. You guys know this, right? What are the building blocks of DNA? Guanine, adenine, thymine, cytosine. Those are your states. Now so you can use these DNA sequences, these sequence of letters, a sequence of characters to build trees. So really, DNA revolutionized not just the study of plants, but the study of all life. You don't know it, but you are living in what will be remembered as a golden era of systematics. Our knowledge of all lineages of life has changed so dramatically over the past 20 to 30 years, all because of DNA and using DNAs to build phylogenies. We've learned more in the past 20 years than we probably learned in the 200 years prior to that. So when people write textbooks in the future, they are going to say, you lived in the golden era of systematics, because we've learned so much. So here, this isn't bold, this is important, this is a test question. Why is DNA so useful? Why not just use these hairs? Why not just use features of the leaves, okay, or the pollen? Here's why DNA is so great. Numerous characters. If every gene, say, has 1,000 characters, 1,000 positions, and there are 20,000 genes in a plant, or in most organisms, you do the math. Am I going to run out of characters anytime soon? I don't think so. All right, so huge number of characters, and this is so important. Another term for it, homology, is so clear. But what do I mean by homology? Similarity due to inheritance of a feature from a common ancestor is clear. I gave the example of wings. Okay, a number of things have wings, but what's the homology of those organs? It's not always clear. Even as something as obvious, you think, as a petal, okay? See these things? We call these petals, right? Well, different flowering plants that have petals, showy things, did not derive all of those petals from the same organs. You can get this in different ways. You can modify a leaf and get a petal. You can modify a stamen and get something showy and pretty and that looks like a petal. They are not the same evolutionarily, but they look the same. But the homology with DNA can be so obvious, so clear. That's why it's such a great thing to use. Here it is. This is a gene called rubisco. It's probably, it is in the chloroplast. It makes an enzyme involved in photosynthesis, a protein. Probably the most common protein in the world when you think about how important photosynthesis is. And here are a bunch of plants, and here's how we've aligned these gene sequences. Look how nice. Isn't that beautiful? You could study this for hours, but you don't have to because the computer will, and will help you generate a tree. All right, everybody good? I want everybody to understand why DNA is so important. So here's a history of DNA sequencing. You don't need to remember a lot of this, but it is amazing to think that the first DNA sequencing was not done until 1975, okay? That's not that long ago, all right? By 1983, another revolution occurred, and this is something called the polymerase chain reaction, which people often just refer to as PCR. How many, honestly, just have heard of PCR? Good, probably just about everybody. What do you do with PCR? Well, I say there, right? What do you do with PCR? This, when you watch any of your CSI, any of these things, there's a little bit of evidence at the crime scene, maybe a drop of blood. How are you going to find out, a DNA match with a tiny drop of blood? You use PCR. You have to make millions and millions and millions of copies of the DNA in that tiny little sample, and then you can get enough DNA to work with. So PCR was a revolution, not just for crime scenes and detective movies, but also for science, because we can use that. I get a leaf fragment from Madagascar. Nobody's ever looked at this plant. Whoa, look how tiny it is. I'm gonna do PCR to make lots of copies of what I want to sequence, and then I can really figure out how it's related to other things. Now, we have moved into a new era of DNA sequencing. By the early 2000s, DNA sequencing just took a huge jump in terms of output with a huge drop in costs. So they called that next generation sequencing. We have subsequently entered a new era where it's jumped in output again and cost has gone down further. So they cleverly call that next generation sequencing. Actually, now I think we're in next generation sequencing because of the new tools, the new methods that come out, really new machines. So here's the PCR PCR polymerase chain reaction. Kary Mullis, who invented this, won the Nobel Prize for this little innovation. And you guys have seen this. I guess everybody knows how to do this, right? You take a little bit of DNA, you pull it apart, you copy it, you pull it apart. By the end of the day, you have a ton of DNA. So this is what I said is used in crime scene. You know, imagine you're in CSI, but this is CSI Botany 2710. And you don't care, you know, you want to figure out how things are related. With 10 more DNA sequences, I think we can nail this phylogeny. So this is a common tool used throughout all areas of systematics. I don't care if you're working on plants, bacteria, you know, viruses, animals, you name it. Everybody is doing the same thing. Okay, everybody good with that?